All right, well, we're going to go ahead and get started with today's Department of Medicine Research Grand Rounds. Obviously, I am not Dr. Williams. He had to make a trip to DC. So today I will be introducing our speaker. So today our speaker is Dr. Parag Shah. Dr. Shah got his undergraduate and graduate degrees in India. I'll tell you a little bit more about that maybe in a little bit. He worked with, as a graduate student, he worked with Dr. Devendra Parmar, who's a well-known researcher around the world. He came to the U.S. To do a postdoc position, originally with Dr. Shan Kakar. And then when I came and joined the University of Louisville Brown Cancer Center in 2011, I was doing a search for a postdoc to work in the lab, and Parag came and was interested in the position. And after some back and forths, we decided to go ahead and give it a try. And he was in my lab for basically ever since then. He's a very prolific researcher, generates lots of data, has performed lots of experiments, published a number of papers. And then about a year and a half ago or so, he moved up the ranks from postdoc to senior scientist to assistant professor on the term track. And so this is one of his first presentations as faculty in the Department of Medicine. So without further ado, I'm going to let Parag take it away and tell you about his research.
Thank you, Levi, for such a nice and kind words and brief introduction. So today I'm going to talk about the protein called Ubiquilin, are the novel tumor suppressors in breast cancer. So a little bit about me. So basically, I'm from the North India. You can see here, this is the central part of North India called Maharashtra. Everybody know that Bombay, Mumbai. And then from here, I did bachelor's of pharmacy, basically my bachelor's, and then moved here, the city, still in North, in Rajasthan. And here I did my master's in biotechnology and moved here in city called Lucknow, where I did my Ph.D. So my Ph.D. topic was related to lung cancer. I basically work on the lung cancer and trying to correlate the genetic polymorphism associated with the cytochrome P450, one of the major metabolizing enzyme in the body, and its association, the polymorphism with the susceptibility towards the lung cancer. So I use like a lot of patient samples, and I used to isolate blood and then DNA, RNA, and try to correlate how we can correlate the SNPs, that is single nucleotide polymorphism within the particular cytochrome P450 family genes, like cytochrome P450 1A1 and 1A2, and then its association with the lung cancer. So I correlated with a lot of environmental factors like socioeconomic status and living habits and like drinking, smoking, what are the different factors associated with that. And then to further my career in 2008, I moved to United States and pursue my postdoctoral training. And so my current area of project, current area of research are basically identifying the molecular and cellular processes involved in the cancer progression and metastasis. Because I had already the lung cancer background, I continued my research in the lung cancer, and eventually recently we started working on the breast cancer and trying to identify the different molecular drivers for the especially highly metastatic breast cancer. And then my major interest of area is the epithelial mesenchymal transition. That is the major step involved in the cancer metastasis. So I will go in briefly little bit. We published number of papers. These are the few examples here on the epithelial mesenchymal transition. So ubiquilin protein has been shown to be involved in the endoplasmic reticulum associated protein degradation pathway. But we were the first one to show that apart from its function in the ERAD pathway, it plays an important role in driving migration and mesenchymal transition, like from epithelial to mesenchymal transition in lung adenocarcinoma cell line. So these were the first study to show that its correlation with the EMT. And then eventually we published another protein called velocin containing protein. It's also called as the P97, which has also shown to be involved in the endoplasmic reticulum associated protein degradation pathway. And we show that the destruction of VCP either with the pharmacological inhibition by the drug like called irastatin or by genetic knockdown, genetic manipulation of VCP, we can induce the ER stress and ultimately induce the EMT. And we also show that ER stress and EMT are reversible processes. And there is a critical balance between ER stress and EMT. Like at some point, like ER stress induce the epithelial mesenchymal transition, but excessive ER stress is bad to the cells. So cells start dying and it causes the apoptosis in the cells. Then another area, like, another paper we published that is the common chemotherapeutics that are used in the clinics, like known chemotherapeutics, including cisplatin, that also cause the EMT. So, and it has been correlated with the ER states, and that is also the reversible process. And so this is the basic, like why metastasis is important, because most of the patient, like any patient, like any cancer patient die due to the secondary metastasis. Primary site, primary cancer, we can easily treat, but once it get to metastasis in distant organ, it is difficult to target and treat. So it is very important to target the metastasis. So this is the basically the schematic of the metastatic pathway. So here the primary site, here the primary tumor is here. So let's say in the breast cancer, and then cells started proliferating to the secondary site. So there is a cell aggressive, like, cell division, more cell viability, and then they try to migrate, they separated from each other and try to migrate to the secondary site through the blood and lymphatic system. And then eventually they try to invade and to the secondary site, and they colonize and form the secondary tumors. So here example, you can see here, the secondary site is lung. And here he started from the primary site here. So there may be the another different site, like it eventually, like if the cells grows, keep on growing and starting, like, migrating and invading into the different distant site, it may metastasize to the bone, brain, and liver. So this is the EMT, so epithelial mesenchymal transition, why it is important. So this is the step. So the term coined in 1960, epithelial mesenchymal transition, by the lady called Elizabeth Hay. 1960, even I don't think we born at that time. Not we, but at least I didn't born at that time. But it was so day by day, it's improving. And like 1969, people have correlated the growth factor association with EMT. Then eventually, in 1994, different transcriptional factors and all kind of stuff. So during EMT, what happened? The primary, like epithelial cells, they try to lose their cell-to-cell contact and cell-to-cell like polarity and try to migrate in the secondary site. So there is a loss of polarity, and there is a gain of invasive and migratory capacity. So during EMT, there is a structural and phenotypic changes or morphological changes. So the cells started forming elongated structure, formation of protrusion, elongated like philopodia, lamidopodia. So this is you can literally see here the cell kind of rounded, and then there is a morphological, because they try to invade into the secondary site. So there are the different proteins that are involved during the EMT. So during epithelial, these are the epithelial marker, the protein expression of E-cadherin, ZO1, that is a occludin family member. The expression is decreases, and there is a expression increase in case of lot of mesenchymal marker like vimentin, cadherin, and alpha smooth methylactin. And there are the pathways that has been associated with the EMT or the SMAD pathway people are trying to target. So I'm going to cover basically the background, material methods, of what are the different in vitro study, in vivo study, and finally conclusion and current project. So breast cancer, why it is important. So you can see literally here on the right-hand side, 1 in 8 humans will develop breast cancer in her lifetime. So this is big. I think this is a huge. And you can see here in Kentucky also, we are on the top. the breast cancer incidence. Here another, this is the 2023 figures. So breast cancer is top, like estimated cases and estimated death. So what are the different factors involved in the breast cancer? So we know that gender is obviously there, age association. Then people have shown that diabetes and overweight, that is also associated, radiation, exposure, also the habit smoking, and this one, genetic factor also, like including the BRCA, like mutations and all. People have shown that it has been associated the breast cancer risk. And now this is the most important factor, hormone. So hormone and receptor oriented, like if the hormones are there or receptor are there, then you can easily target it, like estrogen receptor, progesterone receptor. So there have number of drugs that are in clinics already and under the trial. But let's say like triple negative breast cancer, like it's highly difficult to target it. So so this is basically, see, this is the figure in 2025. We are expecting 320,000 cases of breast cancer. So breast cancer is basically divided into the 5 types, like HR positive, luminal A, luminal B, HER2 positive, and triple negative. So you can see here the prognosis wise. So best prognosis, and if you go like receptor status, if receptors are not there, there is a worse prognosis. So the protein we are talking today, like, our interest of protein, this is called the ubiquilin. So ubiquilin is a 5 family protein member, ubiquilin 1 to 4, that is ubiquilin 1, ubiquilin 2, ubiquilin 3, ubiquilin 4, and ubiquilin L. And it has been shown to be involved. This has an N-terminal ubiquitin light domain and C-terminal ubiquitin associated domain. And it has been shown that involved in the shuttling of the protein to the proteasome during the ERAD pathway. And ubiquilin 1 is basically lost or underexpressed in the lung cancer. And we were the first one to show that apart from function in ERAD, loss of ubiquilin 1 or 2 drives the EMT in the lung cancer. So what about breast cancer? So I was really interested to look like in the breast cancer, and then I started digging the data online, like related to correlation of like receptor status and our protein of interest. So you can see here, the in ER negative status, ubiquilin low is worst compared to if the ubiquilin high expression is there. And this is exactly opposite in case of ER positive. You can see here, in case of ER positive, ubiquilin high expression is worst. So we ask the question, does ubiquilin plays a role in ER negative breast cancer progression? So we have a number of we collected a panel of breast cancer cell line, either like depending on the estrogen receptor status. So these are on the left-hand side, you can see here, these are ER positive and these are the ER negative. And then we have a lot of reagents available in the lab performing the research, like antibodies and siRNA. We use, instead of transient knockdown, we did like shRNA and even like CRISPR-Cas9 gene edits. And different methods I studied for the breast cancer. This one different, like cell viability and all these techniques. So the first experiment we'll trying to see the expression level of expression level of ubiquilin 1 and 2, taking into consideration of the ER status. So you can see here the expression, like in case of ER negative, both ubiquilin 1 and ubiquilin 2 are underexpressed. And then we acquired the so we saw that in vitro, like in the different cell lines. So we were interested, so whether it is real in the tissues also. So we acquired the patient tissue sample. So these are the normal tumor versus adjacent normal tissue samples from the same patient. So you can see here, the N1 correspond to T1 and N2 correspond to T2. We did this one even more tissue samples, but this is just a representation. You can see here, ubiquilin 1 and ubiquilin 2 are underexpressed in the match normal tumor versus the matched normal patient sample. So we asked the question, what happened when we knock down ubiquilin in breast cancer cell? So this is the first experiment. If you remember and recover, I showed you during the metastasis, what happened, cell try to more viability, cell try to proliferate, migrate, and then invade into the secondary site and colonize and form the secondary tumor. So I try to do some in vitro assays, try to study the metastasis. So we plated the cell in 6-well plate, transfect with siRNA targeting ubiquilin 1 and ubiquilin 2 to knock down ubiquilin. And then we recounted the cell after 24 hours of transfection and replated in 96-well plate in triplicate and assess the cell viability for continuously for 5 days using the LMR blue. So you can see here, ER positive, we use MCF7 in one cell line, but we did in multiple. This is the basic representation. So you can literally see here non-targeting siRNA. And when you knock down ubiquilin 1 and ubiquilin 2, so these are the two different siRNA targeting ubiquilin 1 and two different siRNA targeting ubiquilin 2. And this is the positive control of KIF11. It caused the cell cycle apoptosis or cell arrest. So this positive control we always use to show that your transfection work. So, and you can see here, so there is either cells are not growing or cell dying following the loss of ubiquilin 1 and 2 in ER positive cell line. But what happened in ER negative cell line? So we did in a couple of cell lines, same knockdown of ubiquilin 1 and 2, but you can see the dramatic difference between ER positive and negative. Cells start more proliferating, more viability when you lose ubiquilin 1 or ubiquilin 2. Then I was interested to see what happened to the migration when you lose ubiquilin 1 or 2. So same thing, like I did experiment, I plated the cells, transfect the cells after 24 hours of transfection. I made the scratch using the pipette tip, and then you monitor the that gap, filling of the gap, or it's also called the wound migration assay. So the wound closure, you can measure and quantify that after 24 hours and post 48 hours of that cell migration. So literally you can see here, this is the ER negative cell line, and this is the control that is non-targeting ubiquilin 1 siRNA, ubiquilin 2 siRNA after 1 and 2 days, cell almost like filling the gap, like when you lose ubiquilin and 1. So cells started migrating more when you lose ubiquilin 1 or 2. So this is another experiment called clonogenic assay. So you can determine the clonogenic property of the cells. So same experiment I have set up like in the way, and then I transfected cell to knock down ubiquilin 1 and 2, and then after 24 hours of transfection, trypsinized the cells and reseeded 1,000 cells in 6-well plate in triplicate. And after 10 days, still cells and the colonies formed were fixed, and the number of colonies were counted. You can see literally here, this is the control, if 11 cell either die. And you can see the dramatic difference here, but these are the two different siRNA. There is a dramatic difference. There is a much more number of colonies, and the colonies are bigger when you lose ubiquilin 1 and 2 here. And then what about signaling pathway? When you lose ubiquilin 1 and 2. So you can see here, this is the pan-ubiquilin antibody, which take both ubiquilin 1 and 2. The top band is ubiquilin 2, and the bottom is ubiquilin 1. So you can see this is the confirmation of the knockdown. So these are the protein involved in the proliferation. You can see there is a increase in all signaling cascade, increase in expression of the protein. So this is the confirmation of the ER status, like showing that there is no expression of estrogen receptor. And there is like all the dramatic pathway like related to EGFR signaling. There is an increase when you lose either ubiquilin 1 or ubiquilin 2. This is another cell line. So we try to confirm with using multiple cell lines. So now what we saw so far. So we saw that loss of either ubiquilin 1 or ubiquilin 2 increase in cell viability, cell migration, clonogenic potential, and cellular signaling. So we asked the question, can overexpression of ubiquilin block these processes? So for this, we generated the overexpressing vectors So this is the empty vector cherry. By using the lentiviral infection, we generated this empty vector expressing red cherry and then overexpressing ubiquilin 1. So this is the ER negative cell line. And then we look for the signaling cascade. So this is the confirmation of ubiquilin 1. You can see here, these are the two vectors and these are the two clones we selected to pursue this study. And you can see here, it completely blocked whatever previously we saw, that overexpression of EGFR and downstream signaling cascade following the loss of ubiquilin 1. So overexpression of ubiquilin 1. So what about these processes? Like we can see here, these are the two vectors, control, and this overexpression completely blocked the cell viability. What about cell migration? You can see here, it's completely blocked the cell migration. Clonogenic potential. These are the two empty vectors and overexpressing vectors. You can see here, you can completely block all these processes by overexpressing ubiquilin 1. So now everything we saw is in vitro. So what happened in vivo? It is relevant to like happening in vivo also. So whether it plays important role in tumorigenesis or increase in metastasis. So we generated for this one stable shRNA targeting ubiquilin. So these are the two shRNA targeting ubiquilin 1, and this is the control shRNA, and this is expressing GFP. So we confirm that, and this is the knockdown confirmation in ER negative cell line. So the first experiment we set up is subcutaneous injection. This is not exactly physiological relevant model, but you can see here, people try to correlate like injecting subcutaneously, whether they in metastasis to the secondary site like lung. So this experiment we injected, so we use multiple mice in a group and we injected 500K on the right flank of the mice and then followed for 4 weeks. So in between this time, every week we monitor the tumor size and tumor burden. And then following 4 weeks, mice were sacrificed and then tumors were harvested and then protein were collected, and then we performed the Western blot to look for the signaling involved, whether it's involved like in loss of ubiquilin 1, increase in all the proliferative markers. So these are the different mice. See, 5 mice these are the control shRNA. These are the 2 different shRNA targeting ubiquilin 1. So this is the knockdown confirmation. So this is obviously ER negative cell line. There is a ER negative estrogen receptor not present. There is a dramatic activation of these proliferative markers. You can see here EGFR and downstream AKT and ERK signaling. Then we also saw this one like in lung metastasis. This is just a representation. This is the control shRNA and this is the ubiquilin shRNA. 2 different shRNA targeting ubiquilin 1. So you can see there is a dramatic difference between ubiquilin 1 shRNA and control shRNA. So we did in multiple mice, but this is just a representation. This is just a quantification with the image analysis. And here you can see here, literally there is a significant difference. We did repeated experiment this one in a multiple cell line, including triple negative breast cancer cell line. Obviously, triple negative is more aggressive than ER negative, but you can still expect some kind of metastasis. But when you further knock down ubiquilin, you can see there is a dramatic increase in metastasis. Then we were curious about, we saw in vitro like cell either die or cell don't grow. So we did one preliminary experiment using the ER positive cell line, and you can see the difference here. There is no metastasis at all. So either cell die or they don't metastasize. Then the second experiment we performed is a mammary fat pad injection. This is the more physiological relevant experiment using the MDA MB231. So we generated CRISPR-Cas9 mediated knockdown like of ubiquilin 1, ubiquilin 2, and even the combination of 1 and 2, because we saw some additive effect when you knock down ubiquilin 1 and ubiquilin 2. So 50,000 cells were injected in mammary fat pad across. So these were the luciferase expressing cells, and after injection, the tumor growth were monitored for 6 weeks, and at the end of the 6 week, mice were euthanized, and then tumors were harvested. You can see here, and then we can take the images in the CTR building. There is a machine called ME, and you can literally see the difference here. And you can see the progression of the metastasis, like the tumor size. So you can monitor on the weekly basis. So this is the tumor volume, and this is the quantification here. And then when we look for the metastasis, it's a dramatic difference, huge difference. So this is the just the base, or like one representative figure showing that control, and then ubiquilin 1, ubiquilin 2, and then combination of two. You can see dramatic difference in metastasis. So what we observed so far? So loss of ubiquilin 1 and 2 increases cell viability, migration, clonogenic potential, signaling, tumorigenesis, metastasis in vivo also. So I was curious, what is the mechanism behind that? These all the processes like ubiquilin is involved in. So I started looking, and there were a number of papers published related to heat shock protein 70 and the correlation with ubiquilin 2. So heat shock protein has been shown to involve in activated, like, ER stress inducible, and then overexpression of heat shock proteins and its association with all this signaling and all kind of stuff. So I was curious, what about breast cancer? And in one of the paper that showed that ubiquilin 2 interact with HSP70. So I was thinking, like, is it true in breast cancer? So I did one of the experiment to see whether ubiquilin 2 interact with heat shock protein 70. So this is basically like what I did using MD-MB231 cell line, that is a triple negative. I transpected cells with ubiquilin 1 siRNA, ubiquilin 2 siRNA, and then along with the control to look for the interaction. So after 24 hours, after total 72 hours of transfection, I harvested the lysis, and then protein was incubated with the ubiquilin 2 antibody. And then the Western blot performed to look for the interaction for HSP70. So you can see here, there is no band, or in case of ubiquilin 2, indicating that ubiquilin 2 interacts with HSP70. So this is the knockdown. So you're not supposed to get any kind of band here. And then here, there is no ubiquilin 2. So obviously there is a no, and because this is ubiquilin 1 knockdown, and you can literally see the ubiquilin 2 band in both cases. And this is just input. You can see here confirmation of the knockdown, and this is the loading control. And we saw, like we did repeated in another cell line, ER negative cell line, and consistency in the result were obtained. So we use this pan-ubiquilin antibody, which detect both the band. You can see here the top band ubiquilin 2 is not there, and then ubiquilin 2 antibody, that is different antibody, which detect only ubiquilin 2. So confirming that your interaction experiment work, and then you are like, you can literally see ubiquilin 2 interacts with HSP70. So we ask the question, okay, so now they interact. So why this interaction is important? What about other processes? So can we alter the cell viability using the HSP70 inhibitor? So there is a drug called 17AG that is blocks the interaction, or no, not block interaction, but actually that is an exact inhibitor of heat shock protein of different family, HSP70, 90 as well. So we performed the similar kind of experiment. We transfected cells in case of cell viability with the non-targeted siRNA, and then ubiquilin 1 and ubiquilin 2 siRNA. And then before cell harvesting, we treated for 24 hours either with vehicle or 17AG, that is the inhibitor of HSP70. And you can literally see here, when you lose ubiquilin 1 or ubiquilin 2, there is a dramatic increase in cell viability, as we saw multiple times previously. But when you treat with 17AG, you can completely block that cell viability or cell proliferation. So that inhibitor completely blocks that increase in cell viability following loss of ubiquilin 1 or 2. So what about migration? So we set up the same kind of experiment and followed the cell migration, like, the wound closure for 24 hours and 48 hours. So you can literally see here, when you lose ubiquilin 1 or 2, you cannot even see the gap or the scratch or the wound you made with the pipette tip following like 48 hours. But here, like 60% gap is still there. Like, means like you can literally see here, cells migration you can inhibit by using this inhibitor, which is increased by following loss of ubiquilin 1 and 2. So this is the quantification. So what about signaling pathways? So you can literally see, so this is the siRNA targeting, non-targeting, like control, and ubiquilin 1 siRNA, ubiquilin 2 siRNA, either treated with vehicle or that 17 AG drug for 24 hours. So when you lose ubiquilin 1 or 2, so there is a all signaling cascade, like protein involved in cell proliferation, including EGFR, downstream SAR kinase, focal adhesion kinase, AKT, phospho-ER. You can see here there is an increasing all the expression of this protein following loss of ubiquilin 1 or 2. But when you treat with drug, you can literally block this protein, like expression of this protein. And we did repeated experiment in another cell line and we found a consistent result. So now we think that what is our working model? So we think that we saw the lot of protein interact with ubiquilin, like transmembrane domain containing protein, including HSPs, and then we saw like SAR kinase, different kinases, focal adhesion kinase. So we think that it interact with all the protein, like protein X, any interact, not any, like whatever the protein we saw interact with ubiquilin through STI1 and 2 domain in the cytoplasm and keep it inactive. But as soon as you lose ubiquilin, the protein get activated and all signaling cascade and all metastasis and all kind of stuff. So this is our like working model. So my conclusions are basically loss of ubiquilin drives the cellular processes, all cellular processes, including proliferation, migration, clonogenic potential in especially ER negative, including highly aggressive breast cancer, triple negative breast cancer cell line. And all the signaling we can see like activation of following the loss of ubiquilin involved in breast cancer progression and metastasis. And loss of ubiquilin drives the metastasis and in case in Y also we saw. So by identifying and targeting different molecular drivers following loss of ubiquilin, we can ultimately halt breast cancer progression and metastasis. So current projects, I'm basically working on different projects. So these are the recent project, like recently we published this paper. So this is the project, how bidirectional crosstalk between lung cancer cells and microbiota within the tumor can be targeted to combat the lung adenocarcinoma, increase cure rates in the patient. So we published these two papers. There was a graduate student, Alex, he graduated before 1.5 years or 2 years before, and he initiated this project. And then Levi was having the, in fact, he has a collaboration in the Canada, and they came up with this where they did like match patient sample, they collected and did the sequencing and look for the expression different bacteria, and they try to correlate. And then we also did like, what are the different factors, because eukaryotic bacteria cannot secrete essential nutrient inside the cells, but bacteria can secrete, including like methionine, which is driving the, we saw that in aid in like lung adenocarcinoma progression. And we also correlated in this paper, like, if you restrict the methionine, you can inhibit like or block this lung cancer progression. So definitely by blocking this crosstalk, there is a symbiotic association. We can target some of the lung adenocarcinoma. And this is another project. Recently, I started working in collaboration with the Rob in the molecular molding core at the BCC. So this is the nucleolin. So this is the protein, like we saw there is a like inverse correlation, and we saw the overexpression. It has been reported that this nucleolin protein has been overexpressed in the number of cancer and has been associated with the poor prognosis in breast cancer. So we are trying to develop some small molecular inhibitor targeting nucleolin. And this is another project I'm working on, like vidaferin, that is the natural product which can potentially therapeutic agents against the breast cancer. So finally, I want to acknowledge our lab, like we work as a group, like Total Lab. Basically, my major thanks are to Levi and Leah. So I think these are the major pillars of our Total Lab. We work as a group and always help each other and always try to collaborate, and like even though like even the small experiment, like even the like we are sacking or like taking down some mice, like 40 mice, so we try to help each other in the lab and all kind of stuff. And these are the past members and these are the current members, and I'm also thankful to my collaborator Rob, Mike Sabo, Brian, and Doug Dean, who is like expert in case of like EMT, and he work with Zape and all kind of like stuff. And so he has a mouse model for Zape as well. Thank you.